We're going to get started with our next international presentation from India. And Binay is going to tell you about a very exciting new program that's developing there.
Okay. Good afternoon, all. Thank you, Nancy, for the invitation. And I'm going to tell you a little bit about very recent efforts in synthetically producing plant-based terpenoids. I am not a plant biologist, so I don't know much about the plant except the fact that I just love this plant. Okay, so we are a not-for-profit organization started in 2010, based out of Bangalore in India. We don't, so far, except for the recent couple of years, work on SynBio. We're primarily a genome biology lab, working on the genome and computational biology of cancer, specific to head and neck cancer, which is very prevalent in our geography. And I'm going to tell you recent efforts about engineering terpenoids of plants. Now, what are terpenoids? Probably many of you who work on plants know better than I do. They're a very diverse group of natural products, plant-based, and they have applications wide-ranging from being pharmaceutically important to agriculturally important and antimicrobial as well. And our efforts have been primarily focused on a single plant. And the reason this plant is very important because for many thousands of years in ancient Indian history, the plant is known for its diverse kind of activities, starting from useful in diseases to being very useful as pest repellent used in agriculture. And its antefeedant properties of some of the plant compound, and particularly a compound called as a directin. This is a triterpenoid and a very complex chemical, as you can see with 35 different carbons, has a very strong antefeedant activity, and that's exactly what we are after. In addition to azadirachtin, the seed of the plant, particularly the kernel, the part which is inside of the seed, has more than 100 different types of secondary metabolites. But we are only interested in azadirachtin because a lot of biology is known about how the effect of azadirachtin on different types of pests. Different parts of the plant, including leaf, bark, stem, and root, also produce metabolites. But the amount of metabolites produced in these parts are actually very minute in comparison to that of the kernel. Sometimes when you measure that with the HPLC, they are not even detectable, although they are present. And I'm going to show you some of the preliminary data on the relative expression of the genes related to the terpenoid in different organs. This is how they look like. Each terpenoid has a base, what we call as isoprenoid or isoprene. And depending on the number of isoprenes and the number of carbon, they can be classified from monoterpenes all the way to di, tri, tetra, and sesquiterpenes. And the reason we are after this chemical is because there's this paper on Steve Lay, who works in the University of Cambridge as a chemist, organic chemist, and the culmination of a 22 years effort to make this compound as chemically synthesized finally happened. As you can see, there are more than 70 different steps, and it took 22 years. So the total chemical synthesis of this particular product is extremely cumbersome. In addition to the time, the yield at the end of this chemical synthesis is very minute. So we have a problem that even if we have a process in hand, that's totally not scalable. So we need to do something else. Now, in addition to the total chemical synthesis, people also have tried plant tissue culture using suspension cultures, also plant tip cultures. In both of those cases, also the yield has been very low. So it seems metabolic engineering probably is the only viable way if we want to produce a large-scale synthesis or large-scale azadirachtin and other metabolites. Now, we were very encouraged in 2013 after seeing the publication of the metabolic engineering of artemisinic acid. Now, because that's also a terpenoid, and that was produced in yeast, so initially we were thinking maybe this is a project that we shouldn't try and have others try first of a related compound and then enter, because they're very complex. But we were very encouraged in 2013, and that's exactly the reason why we thought maybe it's worth a try. Now, before we even went to synthetically producing in a heterologous system, we sequenced the genome of this plant and also transcriptomes from various different organs. And we're coming from a genome biology point of view to try to understand, because nothing is known about the pathways in this particular plant. We wanted to put the pathways together, understand the important genes that are responsible for the production of this chemical. And also, once we understand the flux, we probably would be able to divert the flux in a heterologous system. That's our aim. Now, before we go into the metabolic engineering, this is a plant which is widely available in India. In fact, the plant was taken from India to different parts of the world, for example, China and Africa, and is in Africa particularly is thriving because it doesn't really require any taking care of. It's a tree. It just grows everywhere, even in a very arid condition without any water. And now the question comes is, before we go into what we call, science 2.0, why can't we just grow this plant? Just take the reason of the piece of land or which is not arable, useless land. Why can't we just grow this plant? Because that seems to be a better and easier solution than doing metabolic engineering. Now, there are problems with that. For example, today, the industry, and there is a huge scattered cottage industry, what we call in India, thriving based on chemical extraction of azadirachtin from this plant, particularly from the seed kernel. And the way it happens today is people, particularly the poor villagers, they collect the seed, and then they collect the seed and sell it in a local market. The issue is scaling is very difficult. And after a few days, in fact, in the second week, the seeds gather aflatoxin. And I already showed you the data that why the total chemical synthesis and plant tissue cultures are not viable. So in addition to getting fungal infection in the plants and not having an organized collection method, there are also issues of the variation. For example, we did a study when we took 79 different districts, and district is an administrative region in India, like maybe what you call as a county. Throughout the country, as you can see, from the south to the north, almost about 3,000 kilometers. And again, from the west to the east, which is not all the way to the eastern coast border, but around 2,000 kilometers, we sampled the fruits from different plants. We wanted to understand if the variation of both the potency and the total amount per kilogram is the same or there is a variation. And in fact, we found that the chemical from here to here are very different, and then from here to here. In fact, to our surprise, there are not only inter-region variability, but there are also intra-region and intra-tree variability. Although the intra-tree variability is not as much as the inter-tree variability. We do not know at that particular point of time why there is so much of variation. So from our study, we have reduced all the factors, all the A's, the geographical boundaries, the metadata, and the rain, the environmental factors. We have now reduced everything, and we know that these are not the ones which are responsible for the variation. We haven't studied so far the effect of genotype, which I think is a very interesting study, and I'm sure that it actually probably is linked. What's the effect of genotype on the content? And remember, there are two things. One is the content. Second is the potency. Just because we have higher amount of metabolite, that doesn't necessarily mean that it has biological effect. So we do not know whether there is an impact of genotype. And the second one, what are the impact of the biotic factors? So these are the two things we do not know at present. Now with this information, we started seeing the real use of metabolic engineering. Now coming back to the pathways that's known for terpenoids, and as I told you, there's a success story of artemisinic acid, which is also a terpenoid. Before that, I just wanted to show you, this is how the immature fruit looks like. It's green. When it matures, it becomes yellowish green, and when it's fully mature, it becomes yellow. And this is how the seed is. Anybody who is a plant biologist know better than I do. The outside, the pulpy part is called the pericarp, and the inside the seed, which is called the kernel. So the maximum amount of this chemical is in the kernel. But interestingly, some of the precursor of azadirachtin is actually present in a very high amount in the pericarp. So maybe the process by which it is changed from an, earlier forms from the precursor to the later form final product is only happening, it's only happening here in the kernel. Now, this is what was traditionally known for last many decades. In fact, only the recent data also suggested that there is very high amount of azadirachtin in later stages of seed kernel. And what I mean by later stages, as it matures from an immature seed, which is stage 1, to a very mature seed, which is stage 5. So now we know that for sure that the seed kernel at the mature stages contains the highest amount of azadirachtin. As I told you a minute back, the pericarp, which is the soft part of the fruit, which is surrounding the seed, contains very high amount of precursor to azadirachtin, but doesn't have the final product. We don't know why. Maybe some kind of regulation is going on there, and we want to find out whether there is a particular type of a gene which is expressed in pericarp, or maybe an inhibitor, because it's quite known for the terpenoid biosynthesis. It's not just the enzymes which produce this metabolite, but also the inhibitor, which plays an important role. Okay, it's not moving to the next slide. Okay, I'll quickly finish. Here, this is basically the pathway for this particular terpenoid. There are two different pathways. One is called the mevalonate pathway, which happens in the cytosol. One is a non-mevalonate pathway, which is in the right-hand side, which happens in the plastids, for example, chloroplast. And the ones that I have circled are the key compounds, particularly the one, the IPP or IDP here to DMAP or DMA-DP here, and then changing it to tritropenoid. Remember that azadirachtin is a tritropenoid, so we're interested in engineering that in the cytosol. Previous efforts to introduce this particular gene into different systems, particularly yeast and bacteria, has been successful in producing some intermediate, although not the final form, because this was for a different terpenoid. Okay. Why am I not going forward? Okay, I'll skip this slide because we don't have much time. So finally, what we have in a draft form figured out the pathways for the production of azadirachtin is starting with oxidosqualene, which is, there are two different ways. One is the ring intact tritropenoid, one is the sesquitritropenoid. And this is one of the major intermediate, what we call as oxidosqualene, going all the way to azadirachtin. And anything which is dotted line is basically the multiple reactions that we don't understand right now. And we're trying to put these pathways together as we speak. So upstream and downstream pathways, again, I'll just go a little. So from the gene expression studies, we did a transcriptomic analysis of different organs. And I'm showing you on the left-hand side of the genes which are overexpressed in fruit, and on the right-hand side, those are underexpressed, and those are the genes only related to the terpenoid biosynthesis. So we know that some of the genes are overexpressed in fruits, but we do not know their role in actual producing azadirachtin. And also we compared the expression of these particular genes in Azadirachta indica compared to other known angiosperms, which are the higher plants. And what I'm showing you here, 4 different plants, the Arabidopsis, and then I think the second one, if I can see, rice or Aiza sativa, then the third one is citrus, and the fourth one is grape. What I'm showing you is all these critical 10 genes in the terpenoid biosynthetic pathway are overexpressed. And the extent of this histogram, or the horizontal bar, tells how much of overexpression is there in comparison to these plants. So if you see a large color of pink, then it's highly expressed in comparison to Arabidopsis and Oryza sativa. So it tells that in this particular plant, we have terpenoid biosynthetic genes which are highly overexpressed. But whether this overexpression relates to the accumulation of the precursor of mevalonate pathway, that we do not know at present. From our initial efforts of doing sequencing of genome, we learned one thing, that the first version of the genome sequencing that we published in 2012 had a lot of errors, not just in exonic region, but also intronic region. In fact, very interestingly, what we found, and I'm showing you 2 different key genes in terpenoid. The single line at the intron, so initial assembly of our genome had a huge intronic region in some of the terpenoid genes. But when we did a version 2 of the assembly using long reads from PacBio, we found that the introns actually much shorter than what initially we thought. And we biologically deduced that conclusion because when we designed primers for those 2 genes, we found that actually what ran on the gel corresponds to the version 2 assembly, not the version 1 assembly. So right now we are correcting the assembly of all the important genes in the terpenoid pathways, design the primers, and individually we'll try to synthesize them. So this is probably one of the final slides. These are the 18 different key enzymes in the pathway that we would like to study further. And as I told initially, understand and divert the flux. And they are divided into 4 different categories: isoprene synthesis, oxidosqualene synthesis, tirucullyl synthesis, and limonene synthesis, finally leading to azadirachtin. The future work is pretty simple: complete the pathway, understand the flux, divert the flux, understand which precursor is critical, understand and isolate the key cytochrome P450, because those seems to be a very key enzyme which is involved in the synthesis of terpenoid, and also understand the functionalization of the cofactors, clone and express those genes, and develop a functional assay, because there is no functional assays at present. Is the chemical, finally, people use HPLC or PLC to identify. We would like to actually develop a functional assay. And one day, maybe in 10 years, this is my dream, is to come up with a terpenosome, having a single chromosome where we can put all these genes, one after another. Each gene or a gene cassette can be independently induced or inducible and can be introduced in a heterologous system, which can replicate into the next generation so that we can produce the required chemical. These are the people who did the work. This is primarily the work of the senior computational biologist, Nirja, and the people in the lab, funding agencies from the government, India. And the inspirations, finally, we are organizing a genome engineering meeting in Delhi in December. The dates are not known yet. All of you are welcome. And I don't know about all of you, but I would definitely welcome my Chinese friends, because if they can talk to our government how to get money, how they do, I would appreciate that very much. Thank you very much.
Okay, hello everybody. My name is Yasunori from Japan. First of all, I'd like to thank all the committee members actually to give me this opportunity. I'll be back here. I'm so happy to be back here. So every time I come to, but this will be my second time, but I come to GP Writes, I feel like family or some friends. So we have some common interests here. That's why we're here, right? So, I feel that my duty here is just to share the passion to the GP Writes from Japan. Okay, I give you 3 topics today. for the last 10 minutes or so. First, nationwide movement. It's a good kind of celebration for us. And then the two kind of personal thing, my department-specific activity and also a proposal to the GP Write flagship project. So I start with the first one. So on behalf of our the government, actually, luckily we are launching a new kind of program, a nationwide program supported by MEXT, our Ministry of Science. I can read this. Culture, education, whatever, and also the a very good thing for us, and also the JST, I have to say that, Japan Science and Technology Agency. So the title of this project is kind of interesting to us, to you also probably, the large-scale genome synthesis and cell programming. And then our director is Professor Shiomi from Keio University. As you may know, he's an expert, one of the leading scientists in the piRNA and also the arm race between the retrotransposon and host. So, and then this project lands 80 years from this year, and then probably that's what I heard. I'm not guaranteed to get this one yet, but 50 or 60 project will be supported for 3 or 5 years, depending on the program you picked up. And the beauty of this project actually is that there are roughly 10 advisory boards, and then I just put the word to everybody, all the advisors, about their expertise. So as you can see, not only the chromosome thing and then epigenetics, but also cell engineering and then virus-host interactions, microbiome, single-cell mix, and then chemists also. So as you notice that the GP Write doesn't push, we cannot push the GP Write project just by genomist, right, or genetist. So we have to combine all different expertise to push the new technologies and then to find out some mysteries in the genome that we don't see yet. And then I got a word from Professor Shiomi through the JST saying that let's keep in touch GP writers. So it seems like he wanted kind of good connections to you guys, exchange ideas. So it's gonna be fun. I move to the second topic related to my department, my university activity. So after I got the information about GP Write stuff, so our department are very interested in making some consortium between industry and academia. And then so we put the name like a genome architect group very seriously, but although the name is GAG. So, and then we have, again, many kinds of experts here from genomics. And then again, it's very important, social governance and ethics. And also chemists. We have very interesting chemists. Actually, we are going to make some technology without a phosphoramidite to synthesize DNA. So we have some idea. And also we have to apply the method. First of all, we want to create a method for the genome engineering, new technologies in the cell first. But we want to apply the technologies to the animal level so that we have to have some collaborations with animal biologists and also proteomics. And as I said in the beginning, this is a consortium, the collaboration with industry. We have very strong collaborators from industry, Hitachi-san and Dentsu, Osaka Sanitary. And then, so as Jeff mentioned in the beginning, luckily, and then it's a great honor for us to join the ST 2.0. So right now, actually two of my students learned the technologies at Jeff's lab, and then now they came back. And then we started the synthesis of the chromosome 4 right now. I hope I can show you the updates next meeting. And then, but this will be kind of a technology transfer part. Of course, we're interested in yeast as a control, but I'm more interested in human genome. But and then, of course, as you notice, we can do many things, right, in the GP Write. So, but we have to have some flagship kind of concept. So that, I just put the flagship concept is GP Write. So for Japanese, the light and R and L pronunciation is very difficult, but my light is okay, right? So, okay, the GP Light project. So obviously, okay, I mean, I'm serious, okay? But yeah, not like, we're not touching the human body, okay? We just want to do that in a cell. And seriously, we have many redundancies in a cell, right? Not only the size of genome and also the gene family and pathway, and then we have basically the 2 chromosomes, right, from mom and dad. So these redundancies must be a very, it's kind of troublesome to control the cells. So that, yeah, we have to make human genome light. So like this, I don't know, we have 46 right now chromosomes, right? But as Jeff did, probably we can combine them or reduce them, or maybe, as somebody said, putting into different pieces, like 20,000 small chromosomes, whatever. And then along the line, I'm of course working on the pilot project you proved. So working on the introns, the retrotransposon, basically removing these stuffs from human genomes in the cell. So obviously we are making tons of the mutant fragments. Each of them doesn't have introns. And then we are creating some technology, okay, to swap the endogenous locus with the fragment in an efficient way. And then once we found that, probably I want to distribute the technology to the older community for free, just open resource, because right now we're facing that the low efficiency of the homologous recombination in human cells, so that that could be facilitated by CRISPR. And then, yeah, so my interest, this is a slide I showed you last time. I'm very interested in the difference in a gene ecosystem in the different species, especially the strong construct between the human and yeast, right? So gene density, the intron presence, many retrotransposons. So it's going to be a good contrast and then good to be compared. So that one of our questions in the GP Light is to see, obviously this one, right? So by reducing the system, can we approach the human system to the yeast system or not? And if there's some boundary or barriers there, that contains some mystery of the genome functional structure relationship. So that's one of the questions. There are many questions that we can apply in this project. And the last, I just want to give you an idea. Last week, one idea came to my mind after I heard the GP Writes flagship project, virus-free infection-free cell. So I just want to give you some proposal by using my current work. So actually, I've been interested in genes. And then this is the current situation of human genes based on the gene code, the latest one. And then this is a number, okay? Basically, link RNA, long non-coding RNA, and then precursors of small non-coding RNA, and then protein genes, right? But as you know, based on the recent papers, people found that the small ORF from the long non-coding RNA, and then they're functional. And also, I've been working on actually another candidate of the small ORF that could encode functional protein there. So this could be the thing you have to think about for recording the ORF in the human system. So yeah, this is just some examples of the microproteins which are encoded in the long non-coding RNA. And also, if you look at the number of the upstream ORF, it's the many, so you cannot ignore. And then, as you know, the ribosome profiling basically show that the many upstreamers are really translated, but it doesn't necessarily show that the protein are stable. But the one with upstream ORFs we are working right now actually translated stably. And then this one is actually also very interesting in the conservation. If you look at the top panel part, amino acid sequence, so from human to turtle and chicken, it's 100% conserved. The length is 59 amino acids, it's very short. But if you look at the nucleotide sequence, there are many substitutions. For example, between human turtle, human chicken, there's 25 substitutions, but again, these are synonymous, meaning the protein by itself will be important. 2 minutes to, okay. And then we made an antibody for the upstream ORF, and then we found that the top, yep, here. So interestingly, okay, this upstream ORF stably translated at some specific region of the brain. In contrast, the main ORF on the same messenger RNA translated in almost equal level in the 5 regions I tested. So meaning actually it have some function, region-specific function. And then I didn't have time today, and I don't want to show that today on this, YouTube. So that, yeah, but I just want to show you that we made a knockout mouse of the upstream ORF, and I got a very interesting phenotype. So that could be one of the first examples of upstream ORF encoding functional proteins. And also we are looking for the second and third interesting upstream ORF, so that yeah, we clone them. Basically, we clone the very high conserved upstream ORF, and then looking at the intercellular localization, and then just showing something. But anyway, so that I hope I gave you my message. So that upstream ORF and also a small ORF on the long non-coding RNA must be the target for the recoding of the codons. So that's it. Thank you very much for your attention.
Good afternoon, everyone. My name is Loon. I'm from the National University of Singapore under the NUS Synthetic Biology for Clinical and Technological Innovation Program. So I'm speaking on behalf of Professor Chang today, and I'll be talking about the synthetic biology in Singapore, and as well as how we're relevant to GP Writes. So our vision for Singapore is to develop our tiny country into a leading synthetic biology hub by bringing together engineering, science, medicine, and social sciences to create a dynamic environment to inspire innovative and impactful research. So Singapore is a relatively latecomer to synthetic biology. So we started participating in iGEM about 10 years ago, but it was only about 5 years ago that it caught the attention of Singapore's government. And since then, they've been very supportive of developing synthetic biology and the bioeconomy of Singapore. So consortiums have been set up to spur collaborations between industry players, research institutes, as well as government agency to promote the advancement of synthetic biology. And just recently, very timely, there's this article in Nature on the plans of Singapore. So if you're interested, you might just read this. So Sin City, as we are known for short, is where I came from, and it's one of the very first research centers in Singapore in synthetic biology. And we have 6 main research clusters centered around the NUS Biofoundry, which houses the automation equipment and analytical instruments to accelerate our work. So we have the biomanufacturing cluster, which does a lot of metabolic engineering to produce commodity and specialty chemicals. There's the environmental remediation, which helps to recover precious metals from electronic waste. There's a mammalian bioproduction cluster that produces biologics in mammalian cells, and biosciences developed to detect diseases as well as to facilitate their evolution. But the most relevant research clusters in Sin City is obviously the synthetic genomics, where the SC 2.0 project is, as well as the therapeutic cells research cluster, in which we engineer probiotics to confer therapeutic properties, as well as to engineer microbiomes to treat diseases. So you might be curious, why is there clinical in our name? And that's because we are actually housed under the School of Medicine, and we are very privileged. We're very privileged to have this unique, to be in this unique position to have close collaboration and partnership with clinicians from the National University Hospital. And currently there's a research program that is training medical doctors into clinician scientists to promote healthcare in Singapore. And there are 5 main areas in this research program. And as you can see, synthetic biology is the only one that is not a disease-directed field, and which shows that the hospital is actually seeing the potential of using synthetic biology for treating diseases. And building on given our close relationship with clinicians, we feel that probably we can use GP Writes in this direction to further understand diseases and disorders in order to find better treatment. And one particular disease that we have in mind is type 2 diabetes. And the reason is that type 2 diabetes is the latest concern of our ministry, because currently we are the second highest prevalence in diabetes in the world. That's not too far behind the US, and it's forecasted that by 2050, the population and healthcare costs on diabetes will increase by 2.5 times. And given that Singapore is ethnically diverse, it is the healthcare trends, the health trends in Singapore are usually representative of Asia, and so this disconcerting growth in type 2 diabetes might be imminent in other countries in Asia, which is a particularly fast-growing area in the world. So in order to win the war against diabetes, we feel that maybe we can use GP Writes to create a cell model to mimic diabetes, and such that we can study the diabetes-related genes and other factors. And we hope that using this model, we can understand the genetic factors that make some people more predisposed to diabetes, and also to understand the variations in the transcriptomics and translational level that might cause anomalies such as impaired glucose uptake and suppress insulin response, which causes diabetes. And by studying of diabetes is in line with our commitment to treat disease with synthetic biology, particularly with our efforts in metabolic engineering. And studies have shown that perturbed microbiome in the body will cause a variety of medical conditions. And in SynCity, we are using several strategies to treat diseases using microbiome engineering. So we are using commensal microbes and making concoctions in order to restore the healthy microbiome. We're also engineering microbiotics to confer therapeutic properties, such as for applications such as anti-pathogen and anti-cancer. And so if we understand type 2 diabetes better, we may be able to find other ways using microbiome engineering to treat type 2 diabetes. So last but not least, we are also involved in the SC 2.0 project, and as you can see here, we are involved in constructing chromosome 15 with Shenzhen SIAT. So we are at our final stage of construction, and along the way we have met lots of roadblocks. And this is basically synthetic genomics 101 course for us, and we've learned a lot from this experience, and we hope that this will help us in our endeavor in our future GP Write projects. So in CCT, we want to do not only just good science, we also hope that our science can be translated into technologies that can be applied to resolve real-world problems for the benefit of mankind. And we have many international collaborators currently that help us make our dreams come true, and we are always happy to have new friends to do greater things with. And finally, that's Prof. Matthew Chang saying hi to everyone, although he can't be here. And thank you for your attention.
So, as some of you may be aware, the UK has put a lot of money and effort into synthetic biology. And synthetic biology in 2012 was identified by the UK government as one of the 8 great technologies. Technologies that, looking forward, the government felt would be important for the UK economy. And as part of this process, it developed a synthetic biology roadmap, which was published in 2012. And 2 of the key recommendations from that synthetic biology roadmap was the need to invest in a network of multidisciplinary centres, to establish an outstanding UK synthetic biology resource, and also to build a UK DNA synthesis and assembly capability. And so that's what they did. So the synthetic biology centres were a £70.5 million investment. There are 6 of them. So we've got at the top there Edinburgh, which is where I'm from. I'm the director of the UK Centre for Mammalian Synthetic Biology. Then Nottingham, their centre focuses on using methane as a source for platform chemicals. Open Plant, which is a centre between Norwich and Cambridge, joining the centre in Cambridge, and they focus on plant synthetic biology. SynBiochem in Manchester focuses primarily on biocatalysis, and then the Warwick and Bristol centres don't have a particular theme associated with them. They do a diverse range of synthetic biology applications. So just very, very briefly, one slide about our centre. So we work on mammalian synthetic biology, and some of the key themes are stem cell reprogramming. We're using a lot of CRISPR, genome editing, and also using synthetic transcription factors to try and program stem cells, with a particular emphasis on neuronal stem cells. And so disease states, we're looking primarily at Parkinson's disease and brain cancers, therapeutics, and also on at multiple sclerosis treatments. We're also developing patterning systems, and this is an example of some of the work by Jamie Davis, where you can use synthetic circuits to form patterns like animal coats. One of the things that's really, really relevant to the GP Write project is the work of Bill Earnshaw, who has been looking at making synthetic hacks. And this is one of the examples of, he's been doing quite a lot of work with that. Some of it was published very recently in ACS SynBio. And we're also doing engineering of CHO cells for biologics production. So as far as the second recommendation was concerned, the recommendation to fund the UK DNA synthesis and assembly capability, a number of genome foundries were funded to the tune of about £18 million. And we've got the Edinburgh Genome Foundry, which I'll tell you a little bit about in a minute. Then there's a Liverpool Gene Mill, there's the Earlham Institute as its genome foundry, and then the London DNA Foundry based at Imperial College. So what I would say is that the Gene Mill in Liverpool and Earlham Institute focus really on technologies more around DNA sequencing and association stuff rather than DNA assembly, whereas the Edinburgh and London foundries are very focused on DNA synthesis and assembly. So this is the Edinburgh Genome Foundry. It's a really highly automated system. It was put together in collaboration with Thermo Fisher. It's got sort of 3 robotic arms. It's probably one of the most complex and automated facilities in the world at the moment. So it was a £5.3 million investment. As part of the investment, we've been developing tools for card-assisted design of DNA. It's really highly automated. We've tried to remove people as much as possible from the assembly process. We have libraries of parts, particularly focused around mammalian synthetic biology, do combinatorial construction and phenotypic analysis as well. So this is a team of the EGF. Hilla Takata, who also is part of the technologies work package for the GP Writes project. She's our foundry manager. Then we've got Jane, an application scientist, and Scott, a technician. And then we've got 3 software engineers led by Valentin. And we found that the software engineering part of it has been one of the key things as far as our foundry is concerned. So in the foundry, we have people design the construct on the computer. Then it's screened for any sequences that might be sort of pathogenic, and if the sequences are okay, it goes to the build process. And we've got a software to integrate the platform and the design. And the assembly protocols that we have automated are based around Golden Gate, Gibson, and Yeast recombination. As part of a test, we've got long read sequences. And then when we've put the DNA into the cells, we can look at the whole range of mass specs for metabolic screening. And for microbial systems, we've got a robolector for microfermentations. And we take the data from that and try and learn from it to learn about the process, the software, and the biology, ultimately. So, as I mentioned, the software has been absolutely crucial to developing the EGF. And we've had to develop software for integrating the robotic platform, to control the robots, to manage files, to develop databases, and the design tools, and integrate the whole lot together. And we've put all of our software out on the Edinburgh Genome Foundry GitHub. So I think there's about 30 different software programs on there now, which are open source, and anybody can have a look and use them. So as far as the GP Writes project is concerned, in the UK we've decided that the best way to take this forward is to develop a synthetic genomics leadership team. And that team consists of Professor Paul Fremont and Dr. Tom Ellis from Imperial College, Patrick Kai from the University of Manchester, and myself from the University of Edinburgh. And so what we're going to do is get together and coordinate UK research in the synthetic genomics space. And so as part of this process, we have interacted with the Sanger Centre. So the Sanger Centre is primarily known as a DNA sequencing facility and has a long-standing interest in genomes. And so at the end of last year, we had a meeting down in the Sanger Centre and discussed with their scientists areas of synergistic interest. And these were the areas of research that resulted from that meeting. So there was an interest from both sides, the people who want to synthesize DNA and the Sanger Centre, in looking at how the structure of chromosomes might influence gene expression regulation. We were interested in genome reduction, genome recoding, genomic architecture, and understanding mutations and what their impact might be. And also had an interesting discussion around the technical discussion, design of data and tools needed for the genome scale work, new enabling chemistry, tools for construction and delivery of big DNA, and requirements for the measurement technologies. These are all things that have already been mentioned multiple times. So the conclusions that we came to in the UK for these meetings at the Sanger Centre are pretty much the conclusions that have been mentioned already. So as part of our process, we're going to organise an International Foundries meeting. This is going to be held at Imperial College on the 20th and the 21st of June. And anybody who's, I think most of you who have got foundries will have been invited to that meeting already. If you haven't been invited, let me know so we can get you invited. So that's to bring together people internationally who are interested in foundries to see what the synergistic interactions are, where the things are different, where we can work together. The other thing we're going to do later in this year is to bring together the UK community for a synthetic genomics workshop. The aims of this workshop are to bring the UK community together in particular to discuss our UK capability and the opportunities for research and industry, look at the global context, discuss the technical challenges, and discuss how we can best interact with the GP Writes project, and primarily to look at how we can get funding for the work. So where are the funding opportunities in the UK? Well, we have 3 major research councils that fund UK science. The BBSRC, which funds the biological research, the EPSRC, which is the engineering and physical research science research council, and also the medical research council. And there's been a big change in the UK funding landscape, literally started at the beginning of April, where these research councils, which were separate entities, are now being brought together under an umbrella organisation called UK Research and Innovation, and that gives us an opportunity for a lot of cross-council funding, which is good for kind of large-scale projects like GP Writes. The other major source of funding in the UK is the charity The Wellcome Trust, and they fund the Sanger Centre. So we're in discussions with all the research councils and also the Wellcome Trust about funding for GP Writes. One thing I want to highlight today is a new opportunity that's just come about in the last couple of weeks. So the BBSRC, the UK Research Council, and the NSF have come together to make the opportunity basically for UK research scientists to work with US scientists. And so the way that this opportunity works is that we can apply together either to the BBSRC or the NSF. And if it gets funded by the BBSRC, then the NSF will fund the US component. If we put funding in by the NSF and it gets through the NSF review process, then the BBSRC has promised to fund the UK component of the project. So it's a big opportunity. There's a deadline, which is the intention to submit deadline, which is on the 12th of June, on the full application. That's just so that they know how to do the review process. And then the full applications deadline is on the 25th of September. So if any of the US colleagues here are interested in putting a funding application in with UK scientists, let me know and I can talk to you and tell you a little bit more about how that may work. The usual standard funding for the BBSRC grants go up to about £2 million, just to give you sort of a ballpark figure. That's all I have to say, so that's where we're at, and please talk to me if you're interested in working with UK colleagues. Thank you very much.
For the UK DNA construction foundries, how is the relationship with industry in terms of, say, what do you rely on industry for and what do you prefer to do in-house? Is there a particular kind of size transition there, or is it more case-specific than that?
It's case-specific, so we don't make oligos. So we would buy oligos. What we tend to do is buy gene-scale pieces of DNA and do the assembly. So all the foundries do assembly. We don't do just oligos. It's not worth the money for us. It's too much hassle, and it's much cheaper, much easier for us to work with the industry. The other thing is, these are academic foundries. You know, one of the things about the funding of these foundries is that we're not allowed to compete with industry on any level. That because the government is not funding us to compete with industry. We want to stimulate the economy, not compete. And we couldn't possibly anyway, realistically.